Hello everyone, I'm Sashank. I'm from College of Engineering, Indiana University. I'll be presenting a paper, Interrelating N-gram based protein sequences using LSTMs with parallel capsule learning. It is co-authored by Samya Sanadwaru. Genome data is highly dimensional, complex data that demands effective data management policies for high-performance results and to develop efficient solutions. This involves analyzing the interaction of proteins, and this helps to study cell cycle and gene expression regulation, along with energy and substance metabolism processes in living bodies. This helps us to understand the role of proteins and their functional connections in biological systems. In order to identify the relationship between proteins, we go with an embedding-based system, where each protein is represented as a token or an N-gram. These are vectorized using embeddings and fed to LSTMs or long short-term memory. LSTMs help to understand the relation between static spatial features and dynamic temporal features, and they use this relationship to reconstruct the output sequences. LSTMs relate the embedded N-gram sequences to identify commonalities between the input protein sequences. Once the LSTMs extract the embeddings, we then feed these to capsule neural networks. These use dynamic routing algorithm to relate vectorized features which are extracted by convolutional filters in each capsule. By using multiple parallel lanes, we hope to reduce the time taken to train and to increase the accuracy of the model. This is because the network learns features in multiple ways, in each lane, and this makes the network more resilient. System design. So this is a system design where we take protein interaction pairs and create sequences which are N-grams. Each sequence is then tokenized, randomly paired and embedded and fed to an LSTM feature extraction module. From the LSTM feature extraction, there are two parallel lanes which lead to the capsule network. Each lane is responsible for extracting features in a particular way from the same input. This results in the network learning features in multiple ways. Finally, the outputs are correlated by using a dynamic routing algorithm, and we try to make the output prediction. The dataset that we have used comprises of protein-protein interactions from the database of interacting proteins. Each protein pair have less than 50 amino acids that are removed, resulting in a total of 17,257 positive interaction pairs. The negative interaction pairs are generated by pairing proteins from a particular subcellular location with proteins from other locations. This is done to match the 17,256 positive interaction pairs. Once this is done, we create protein sequences which are recognized as sequences of 20 distinct amino acid alphabets. The raw sequence of amino acids are textual strings, and hence NLP techniques can be used to extract features from them. N-grams is one such technique that represents the n-constitutive elements in a given sequence. Each N-gram represents a protein motif, which is a shared subsequence of amino acid. Once this is done, the sequences are considered as an individual token, and this is assigned a scalar value. The number of unique N-grams, which is P, is given by equation P equal to 20 bar N, where N is the order of the N-gram. This gives the total permutations that are present for 20 amino acid alphabets. LSTMs help in modeling long-term dependencies in the given sequence by using memory cells. By identifying sequences and relating them, we can get a temporal dependency between each sequence. Once this is done, the fixed length vectors obtained from the LSTM modules are passed to a capsule layer. The first step is performing convolutions of the given vectors in order to extract features using filters. Once this is done, we pass the output through a squashing function. This squashing function is a nonlinear activation function that reduces the given vectors to a length between 0 and 1. This assigns a relative importance to the feature vectors, as vectors with lengths closer to 1 have more weightage than vectors with lengths closer to 0. The squashed vectors extracted from the input sequences are concatenated together and fed to a dynamic routing algorithm. By optimizing the bias matrix during training, the vector activations between capsules are determined. The final capsule layer has the same number of capsules as the number of classes of prediction, and the probability of prediction for each class is obtained by calculating the root mean square of the activation vectors. Implementation, the setup is performed on Google Colab. So the total number of instances for each of the train, test, and validation set is given in table 1. The number of positive interactions and number of negative interactions are relatively the same. We split the data set into a train test validation split of 74:1:25. The sequences of proteins are concatenated and converted into N-grams with a length of 3. The sequences are tokenized to a maximum length of 1000 using Keras's preprocessing tokenizer with a pre-padding of sequences to ensure that all sequence lengths remain the same. The sequences are then given to a Keras embedding layer with L2 regularization and a weight decay of 0.001. So this is the overall neural network architecture that we have built. This comprises of two layers, two input layers, each consisting of a single protein motif. These are from these we create an embedding layer, which is common for both the sequences. And then we create a batch normalization layer and feed it to LSTMs. These LSTMs are responsible for extracting the initial temporal data from the given sequences. Once this is done, we pass it through two convolution layers which act as filters and extract features, and then it is passed through two primary capsules before being sent to the capsule layer. This capsule layer performs the dynamic routing before being sent to the output layer for prediction of probability. Training parameters. The implemented model is trained for 20 epochs with a binary cross entropy as loss function, RMS prop as optimizer, and a learning rate of 0.001. RMS prop helps avoid monotonically decreasing learning rates and uses a weighted moving average of gradients to update the weights. Binary cross entropy calculates the distance between the prediction and the actual value, and based on that distance, penalizes the probability of prediction. By using a callback function with reduced LR or plateau, the learning rate is periodically decreased by a factor of 0.1, and there is no change in validation loss. Results and analysis. The testing of this is shown in this figure. Most of the PPI instances present are classified correctly into positive and negative classes. The performance is also evaluated using accuracy, precision, recall, and reference score. We can see that the model has a high reference score of 0.92, which indicates a balance between precision and recall. The analysis can be also done in terms of TPR, FPR, TNR, and FNR. And it can be seen that the model exhibits a very low false positive rate and a very low false negative rate. And this indicates high performance of the model for classification of PPI interactions. The AUC value of the parallel capsule-based LSTM network is high at 0.96. We can see that the ROC curve indicates a balance between TPR and FPR, resulting in a better performance of the model. We also compare the model with other state-of-the-art architectures which have used the standard PPI dataset for yeast. It can be seen that our model outperforms the other architectures with an accuracy of 92%. Conclusion, we identify protein-protein interactions by converting the sequences of proteins into N-grams by using NLP techniques. These sequences are tokenized, vectorized before feeding to LSTM cells for extraction of features for classification. The output sequences from the LSTM layers are further passed along to parallel capsules, and this helps to identify the co-localization of the extracted features. We use dynamic routing algorithm that helps to determine the spatial location of the vectorized protein motifs, and finally, try to predict the interactions. The complex features such as amino acid composition, dipeptide composition, quasi-sequenced order descriptors, etc., can also be analyzed for a study of PPI. The expansion of the deep neural architecture can be done using an encoder-decoder network to generate protein-protein interaction sites which are exact in nature. We thank the chairs of CCWC 2022 for giving us an opportunity to present our paper, and we are welcome to any questions. Thank you.